In this video, we will discuss about post-translation translocation in Eubacteria. In the previous video, we discussed this post-translation translocation in mammals. This time we will discuss in Eubacteria. So, what is the difference? Main difference is that in mammals, the complex that form at ER membrane is SEC61, whereas in Eubacteria, the complex that form is SEC-Y. So now let us go into the main topic. So in this case also, the translation occurs in Eubacteria in the cytosol. And this is the signal sequence. So in the last video we have discussed that because of less hydrophobic signal sequence, it is not able to detect it, the SRP, the signal recognition particle, not able to bind to this signal sequence at the time of translation. So in the whole translation occurs in the cytosol. After that, SEC-A complex. SEC-A complex comes and bind to this polypeptide chain. And translocate it to the ER membrane. In ER membrane, there is a complex that is SEC-Y, and this SEC-A comes and bind to this SEC-A, and polypeptide chain comes and bind to this SEC-Y in a manner that, and this is actually is in the form of like some kind of groove. This is SEC-A, this is SEC-Y, and this is polypeptide chain. So when this SEC-A complex binds to SEC-Y along with this polypeptide chain, a loop is formed, and that comes, and part of the loop comes in the ER lumen. And now what happens is that this groove will open up and moves away from the polypeptide groups. Group opens and move away from the polypeptide binding site to the upper polypeptide binding site. Opens and then it moves to the other side. It moves to the upper side of the polypeptide chain. And what happens is that after it binds to the another site of polypeptide, it will then moves close to this complex, SEC-Y complex, and this case and this process continues till the whole polypeptide chain is translocated inside the ER lumen. This is ER lumen. So what the final process occurs? Finally, this whole polypeptide chain will come this groove will now be free to act on another polypeptide chain. So, and during this and after this step, there is further processing of the polypeptide chain occurs to remove this single sequence and convert this immature protein to the mature protein, so that it can acquire its native structure based on the sequence of amino acid and perform the function for which it is been secreted or produced. So this is how the post-translation translocation occurs in Eubacteria. So this is the third type of translocation that we are discussing in this video. Thank you for your time.